Hi guys, so I have started with obstetrics. So from today we'll study obstetrics for a few days and then we'll move on to the next subject. And at the end of this podcast, I'll give you some extra information or something different which is not related to obs, which I have studied. Okay, so let's start from the basic. So in the fetal life, we'll see gonads. So in the fetal life, gonads are initially, for the first 6 weeks, the gonads are bipotential. So they can either convert into testes or ovaries. So what determines this? Whether they'll form testes or ovaries? There's a gene called SRY gene, which is present on Y chromosome. Okay, so if this gene is present, testes will be formed. If this gene is absent, ovaries will be formed. Okay. Now let's see about spermatogenesis. Spermatogenesis starts from a cell called spermatogonia, which is a diploid cell. Okay, this spermatogonia undergoes mitosis and forms primary spermatocyte. So since mitosis is taking place, primary spermatocyte is also diploid. Okay, this primary spermatocyte undergoes meiosis 1, and forms secondary spermatocyte. Okay, so secondary spermatocyte will be haploid. This secondary spermatocyte undergoes meiosis 2 to form spermatids, and these spermatids undergo a process called spermiogenesis, and through this process sperms are formed from spermatids. Okay, so basically two cells, which are spermatogonia and primary spermatocytes, are diploid, and secondary spermatocyte, spermatid and sperms are haploid. Okay, so if I ask you one question, how many sperms are formed from one primary spermatocyte? The answer is easy, 4. But how many spermatocytes are formed from one spermatogonia? So that is 64, because one spermatogonia forms 16 primary spermatocytes. Okay. Now how much duration does this take? Does this entire process of spermatogenesis require? It's around 70 to 80 days. To be more specific, it's 72 to 74 days. Okay. Now let's compare some parts of sperms and spermatids. So nuclear material is present in head of the sperm. Okay, mitochondria is in the middle piece, and in the tail there is centriole. Okay, and in the acrosomal cap is Golgi ball. So you know that only the head fuses with the ovum. The middle piece and the tail does not. That's why all the diseases which are mitochondrially inherited, they do not come from the father. They only come from the mother side. Okay. Now let's see about oogenesis. Oogenesis is slightly different from spermatogenesis. So spermatogenesis starts after birth, and to be more specific, at puberty. Okay, but oogenesis starts before birth. There is similar to spermatogonia, there is oogonia, which undergoes mitosis and forms primary oocyte. This primary oocyte starts meiosis 1 but gets arrested in the prophase. In the prophase, in diplotin stage. Okay, so this primary oocyte is arrested in the diplotin stage, and it remains arrested till the female attains puberty. After she reaches the puberty, there are hormonal changes and LH surge is there. Okay, and basically due to these hormonal changes, this primary oocyte again resumes its meiosis 1, which was arrested in prophase, and this meiosis 1 is completed. So after completion of meiosis 1, secondary oocyte is formed and a polar body is formed. So polar body is it just has nuclear material 23X. It does not have cytoplasm, or it has a very scanty cytoplasm. Most of the cytoplasm and most of the organelles go into the secondary oocyte. Okay, this secondary oocyte again starts meiosis 2, but this time it gets arrested in metaphase. Okay, and during LH surge, when ovulation takes place, this secondary oocyte, which is arrested in metaphase, is released from the ovary into the fallopian tube. Okay, and in menstruation, this secondary oocyte will be shed as it is, unless fertilization takes place. If fertilization takes place, then this meiosis 2 will be completed and ovum will be formed. Okay, Now some important points about oogenesis or ovaries. So when the female is 5 months in the intrauterine life, when she is around 20 weeks old, she has maximum number of follicles, which are about 7 million. Okay, as a few weeks pass by and when she takes birth, these follicles reduce from 7 million to around 1 million. Okay, and till puberty they are just 4 to 5 lakhs. Okay, so if you calculate how many menstrual cycle does a female have in their lifespan, it's around 400 to 500. Okay, so only 500 follicles are released. Okay, in each menstrual cycle. In the entire life, in the entire lifespan. So out of 4 to 5 lakhs, only 500 are used. The rest undergo atresia. Okay. So now this was about oogenesis. Now let's consider that fertilization takes place. So what will happen? So for fertilization, there are some important points. First is capacitation of sperms. What is capacitation? Capacitation is the ability of the sperm to fertilize the ova. Okay, and where does this capacitation take place? It takes place in the female reproductive tract, specifically in the fallopian tube. Okay, so if they ask you where does the sperm become, at which site does the sperm become motile? So that site is epididymis. But where does the sperm become hypermotile, or where does the capacitation takes place? The answer for this is fallopian tube. Okay, so when the sperm reaches the fallopian tube in the ampulla, there is fertilization. So three main reactions take place. First is acrosomal reaction, next is cortical reaction, and lastly zona reaction. Okay, after all these reactions, the sperm fertilizes with the oocyte, and the zygote is formed. This zygote multiplies and there are 2 cell, 4 cell, 8 cell stage, and finally after 8 cell stage there is 16 cell stage. This is called morula. Okay, so this entire process, and then this morula from the fallopian tube comes into the uterus. Okay, on which day? On the 4th day after fertilization, the morula comes into the uterus. Okay, after and still till the time this morula is in the fallopian tube, the zona pellucida is still there because it prevents the zygote from getting implanted elsewhere. Okay, so after it reaches the after it reaches the uterus, body of the uterus, the zona hatching takes place, and now this morula is ready to get implanted. Okay, but till this time the 16 cell stage is also getting multiplied, and from morula blastula is formed. Okay, and the entire structure is called blastocyst, right? And what exactly is implanted in the uterus, in the endometrium of the uterus, is blastocyst. Okay, so now this was till the process of implantation. Okay, next further process we'll discuss in the next podcast. So now let me give you some extra information. So today I read up something about telomere and telomerase. So telomere are some nucleotide repeats which are present at the end of the chromosome and which prevent two chromosomes to get fused with each other. Okay, but the important point is after each division the length of this telomere goes on decreasing, and initially it is around 11 kilobase pairs at birth, and as we grow old it comes down to around 4 kilobase pairs. Okay, but these telomeres are important for the cell to be alive and for the proper functioning of the cell. And there is one enzyme called telomerase. Okay, so what this enzyme does is whatever telomere has been cut off or truncated during the cell cycle, it again forms those telomeres which are lost. This is done by enzyme telomerase. But in some cases this telomerase enzyme can function more. So this helps the cancer cells to proliferate. See, we know that cancer cells are rapidly multiplying. So when they are rapidly multiplying, the length of the telomere will go on decreasing, go on shortening, and ultimately this cancer cell can die. Okay, but if the activity of telomerase enzyme is very high in those cancerous cells, those telomeres, the telomerase enzyme will reform those telomeres which are lost, and thus it will help in survival of those cancer cells. Okay, so this was something about telomerase and telomere. So that's all about today's podcast. Tomorrow we'll continue ahead. Okay, sure. Bye.